On behalf of the faculty and staff of the UCLA Health Ethics Center, I welcome you all to our annual Weinberger Vermut Genetic Ethics Lecture. Before introducing this year's esteemed speaker, let me take this opportunity to renew once again the Center's sincere gratitude to Tom and Leslie Weinberger, who so generously sponsor this annual event and who have unstintingly supported the mission of the Ethics Center from its inception to the present day. Tom and Leslie, from all of us at the Ethics Center and at the health system, a very, very sincere thank you for all you do for us. Now it is both my privilege and my pleasure to introduce this year's speaker, Mr. Vence Bonham. Mr. Bonham is the Acting Deputy Director of the National Human Genome Research Institute and an Associate Director of its Division of Intramural Research's Social and Behavioral Research Branch. Mr. Bonham also leads the Health Disparities Unit, which investigates the equitable integration of new genomic knowledge and precision medicine into clinical settings. Before his arrival at the Institute, Mr. Bonham was a tenured faculty member at Michigan State University and with appointments in the colleges of medicine and law. Mr. Bonham is the author of over 100 peer-reviewed journal articles and book chapters. His research and scholarship focuses primarily on the social implications of new genetic knowledge, genomic knowledge, including the role of genetics in exacerbating or ameliorating racial and ethnic health inequalities. His research group studies sickle cell disease, a condition that has faced significant health disparity and could be further impacted by disparity in access to emerging curative genomic technologies. Recently, the National Academies of Sciences, Engineering and Medicine issued a report on using population descriptors in genetics and genomics research. It is in that context that Mr. Bonham will speak to us about the ethical, legal, and social implications of both the historic and the current use of population descriptors in genetics. He will also speak about the challenges and opportunities to advance health equity in genetics and genomics research. At the end of his lecture, there will be an opportunity to put questions to Mr. Bonham. Given the virtual format we are using, can I ask that you write any questions you may have for Mr. Bonham in the chat, and our senior fellow, Dr. Max Kramer, will ask the questions at the end of the presentation on your behalf. Mr. Bonham, it is my privilege to invite you to give the 2024 Weinberger Vermut Genetic Ethics Lecture.
Thank you. Let me open my slides here. All right, am we in full screen? Great. First, I just want to say thank you for this invitation to give this important, honored talk today. It is a great privilege for me to be here and to participate. Again, I'm Vence Bonham. I am Associate Investigator and the Deputy Director at the National Human Genome Research Institute. And today I really want to explore an area that I think is really important that's at the intersection of bioethics and genetics and genomics research that's important to our understanding of study design and how do we interpret information with regards to genetic similarity and genetic variation. So I titled my talk, Advancing Health Equity in Genomics Study Design, because we're really at a point now where we can make a difference with regards to how we think about descriptors and use them in genetics and genomics research. But let me go back over the last 20 plus years and to just highlight some of the exciting things that are happening and have happened within the field of genomics. The National Human Genome Research Institute led on behalf of the federal government in the United States and working globally the Human Genome Project. And now in 2003, the first human genome was sequenced. And this really set the platform for all of the work that has gone on over the last 21 years that within the field of genetics and genomics that is now important to clinical care and continuing of research within genetics and genomics. The cost of sequencing a human genome has been reduced more than 1 million-fold. This really has been quite dramatic to think about the original costs of more than $1 billion to sequence the initial human genome and what you can sequence a genome today. And how that information is now being used both in research and clinical settings. Millions of humans have had their genome sequenced, either in a research setting or in a clinical setting today. So we've moved really from a perspective of this only being exclusively a part of research to now part of clinical care. The profound advances in understanding of how the genome functions has occurred over the last 20 years. We are learning more of what the meaning is of these variants and understanding how it's important with regards to risk of disease. But there's a lot more that we must learn to fully understand the human genome. And there has been significant advances in unraveling the genomic basis of human diseases. We have learned a lot, but there's much more to go. But what that means is that the field of genomics is now part of biomedical research across disciplinary areas, and the field of genomics is now a part of clinical care. And finally, there are now vivid examples of genomic medicine now emerging, where genetics and genomics and this new understanding and new knowledge are actually impacting care. So my research lab, as was stated, is involved in one of those areas, and that is the treating of sickle cell disease. We've learned a lot over the last number of years with regards to understanding the role that genomics and particularly the issue of gene therapy can play with regards to curative or transformative treatments for sickle cell disease. This graph from John Tisdale and his colleagues identifies examples of treatments that are now available, both from medication to allogeneic transplant to ex vivo gene therapy. In December of 2023, the Food and Drug Administration approved 2 new gene therapy treatments, one a gene editing and one a gene addition for sickle cell disease. And there is significant work going on today about in vivo gene therapy. So we are learning and now implementing this information within clinical care. And this truly comes out of all the work over the last 20 years within the field of genomics. But one of my colleagues, Professor Michael DeBon, who's a professor of pediatrics and medicine at Vanderbilt University, stated when we were both giving talks at Children's National Hospital in Washington, D.C. last April. He said, until we have more data, we will be guessing as to the optimum curative therapy for children and adults with sickle cell disease. And I highlight here, until we have more data, we will be guessing. This continues to be an issue for the field. This is an equity issue with regards to how actually genetics and genomics will be used in clinical care and who will truly benefit from the new technology and the new knowledge that's occurring. So I want to start my talk by focusing in on one of the equitable principles and issues that I see that's important for this intersection between bioethics and genomics, and that is the lack of diversity in genomic databases. We are today lacking significantly the rich diversity of our global population in genetics and genomics research. Our institute in 2020 published a strategic vision, and this strategic vision really lays out what our institute sees as the most important priorities for research and what is the future areas for individual scientists to be thinking about with regards to where we need to be doing more work and more research. And we included a set of principles that we saw as important for the field of genomics. And I want to highlight one of those principles, that we must strive for global diversity in all aspects of genomics research, committing to the systematic inclusion of ancestrally diverse and underrepresented individuals in major genomic studies. This is a challenge for our field. This is a challenge for the ability to truly make sure that genomics benefits everyone, that need more data, we need more individuals involved from diverse ancestral backgrounds. As early as 2009, researchers were asking questions about the diversity of participants in genomic studies, starting with the GWAS catalog, which is the Genome-Wide Association Studies catalog, which is a catalog that has data about various GWAS studies that have been conducted. The original individuals that looked at this included Carlos Bustamante, who looked at the work of what was in GWAS and looked at the ancestral backgrounds of individuals in 2009. And he found in 2009 that more than 95% of all the participants in GWAS studies was from European ancestral populations. In 2016, my colleagues, Alice Popejoy, who is a assistant professor at University of California, Davis, and Malia Fullerton, who is a professor at University of Washington, replicated that work. And when they replicated the work, they saw a change had occurred, that by 2016, only 78% of the participants were from European background. But the vast majority of individuals who were not of European background were primarily from studies from China that were conducted and were not part of the catalog's diversity of global populations. This slide just highlights here some of the major issues with regards to the ongoing challenges that we have with regards to global diversity in these studies. Sarah Tishkoff and colleagues in 2019 looked at, again, the GWAS catalog, and they looked at both the studies and the targeted populations in the studies and the actual participants. And you see here in this figure that they clearly had been a change with regards to having more studies that were seeking to have more diverse populations. But in reality, we were still stuck with this 78% of the participants were from European backgrounds. And as you think about the global diversity, that truly is a minority of the ancestral backgrounds globally. So this has clinical impact. And we're learning more and more about the clinical impact of the lack of participants from diverse ancestral backgrounds. And we're learning that without that information, we don't know if variants are pathogenic or not with regards to individuals. And we have many more individuals that receive results of unknown significance, where we are concerned that we really can't provide quality information with regards to that individual and their risk for disease and what the variants are telling us with regards to their health. This issue has become a major issue with regards to the development of new polygenetic risk scores. This work is ongoing work. Our institute is funding much work in this area, but we've clearly found that the accuracy and the ability to use polygenetic risk scores are much better in European populations than any other populations globally. And that from a implementation for clinical care, this continues to be a major challenge with regards to using these polygenetic risk scores and these tools in clinical care, and how do we really advance this within research? But there is work going on to expand this. The recognition that we need to create new tools, new strategies to have diverse ancestral populations, to be able to use this information, to conduct small cohort studies, various strategies to enhance our ability to use this genetic and genomic information for all people and not just some. Our institute at NIH and other parts of the federal government are working with institutions across the globe to actually create much larger databases, the sharing of data in a way so that we can truly have better knowledge and understanding with regards to risk of individuals from different populations. This map just shows examples of where there are studies that are going on and databases that are being developed, but you can also see that there are major areas of the world where there's no activity whatsoever. And I particularly highlight sub-Saharan Africa and India, but you can see that there are areas that are just missing from participation in these global consortiums. So there's a much to do here, and we are learning more, but we must recognize the limitations that we have today. Limitations where we can't provide adequate and appropriate information to populations with regards to variants, to understand whether a variant is pathogenic or not, because we just don't have enough information. And the issue with regards to our ability to look clearly across the world. So in one of the articles about the need for a human pangenome reference, it stated, broadening the reference to represent genomic diversity and allelic variation within and across human populations will improve variant detection and functional annotation of genomic variants, and ultimately will be transformative for biomedical research. So there's much work that's needed. The National Human Genome Research Institute is leading an initiative to establish a new human pangenome so that there are reference databases across different ancestral and diversity backgrounds to have that information so that physicians can use that in clinical care and that researchers can use it in their work. So I'm going to shift now to focus in on the history of population descriptors in genetic and genomics research. And I want to talk about this from the perspective of why this is important to today in 2024. And I'm going to give you my take-home message on this at the beginning. Population descriptors have changed over time. We must recognize that these are not discrete biological groups that have not changed, but that they clearly have changed based on social and political context. My research lab conducted a study that we looked at the American Journal of Human Genetics, which is one of the leading human genetics journals. And we looked at the history of the journal from 1949 to 2018. This project was led by one of my fellows, Yinji Julia Bayan, and we worked in collaboration with the National Library of Medicine and my colleague, Dr. Lawrence Brody, at the National Human Genome Research Institute, to study the corpus of this journal and to look at how descriptors had been used over time. And so this was a big data textual computation analysis with the journal to understand the changes and the concurrence with regards to various other terms. This is one of the tables from our study, and as you see here, it highlights different types of descriptors that have been used over time and how they have changed and whether they have increased or decreased over time. But I want to really just focus in on three areas. The three areas that I want to focus in on is ancestry, ethnicity, and race. We found in our study that the use of ancestry as a descriptor for populations in genetics and genomic studies increased over the time of the journal from the beginning in 1949 through 2018. Ethnicity increased, but we also saw a decrease in the use of race over time. If you look here, you see the blue line is the number of articles and the red line is the number of segments. So why did we look at segments? So these are like 100 words within the article. We wanted to do some analysis to also look at co-occurrence of words within the articles. And so we looked both at the articles and the actual frequency, but we also looked at co-occurrence with other terms. And we found in our study that there actually was an increase over time between race and ethnicity, co-occurrence of these terms being used, that race and ethnicity were used in the article together. And I think this is important as we think about interpretation and descriptors of the combination of ethnicity and race. And what does that mean? And how do we understand and interpret that? And that this is an important challenge for scientists when they are communicating the findings of their study. So in closing with regards to this study, from 1949 to 2018, the term race has declined in use while increasing in co-occurrence with ethnicity. And at the same time, the use of ancestry and ethnicity had increased. We report that temporal changes in the use of specific population descriptors, adding support to the long understanding wisdom that population labels are not based on immutable biological order, but shift in tandem with social context. I encourage you to read this article and this work that we're doing. Other researchers need to explore and study the question about the history of how different descriptors, different information has been used within the scientific literature over time. And hopefully this will give you something to think about with regards to your own work. So advancing the rigorous responsible genomics study design and population descriptors is essential for us to truly move to what I would consider the new era of genomics and a recognition of the history of the conflation of race and genetic ancestry. This article by Benz and colleagues does a nice job of talking about some of the what I would describe as pain points or decision points that occur within a research study process where you need to be thinking about issues with regards to how our terms, how our descriptors being used. Here it highlights the first decision point is in participant recruitment and enrollment. How do you enroll individuals? What reporting do you use? Do you use your inclusion and enrollment report terms, or do you use other terms to identify your populations? But making intentional decisions in how you describe your populations. Second, stratification of allele frequency data. What's the process? What tools do you use to look at these issues and to look at difference? Interpretation of variance. How do you talk about, how do you communicate variation and genetic similarity when you're talking about variant interpretation? And finally, with return of results and clinical translation. How do you communicate what does this mean from a potential clinical impact and clinical importance? The issue with regards to relationship between genetics and self-reported ancestry is also important as we think about how people identify themselves and how we look at their genetic information and the relationship that there and the relationship that doesn't exist. So I know everyone has seen these principal component analysis that look at variation, and it's clear in all of the work that's been done within the field of genetics and genomics over the last 15 years that we need to understand that genetic similarity and genetic variation is a spectrum. And so as you look at these individual participants who self-identify as within different population groups, there's still a true diversity with regards to their own ancestral background. They're not all the same. And so when we talk about difference and we talk about similarity, we need to recognize and use nuance when we talk about difference within populations. So this is going to really take me now to the National Academies report. The NIH was pleased to sponsor this report. 14 institutes, centers, and offices sponsored this report that was conducted by the National Academies. I was honored to be involved in helping to lead this on behalf of NIH. And this report that came out in March of 2023 is having a significant impact within the field. So the overall review of the recommendations, there was 13 recommendations that were part of the report that came from this consensus study. They were divided into three areas, for general audience, kind of specific requisites, guidance for researchers. There was guidance to help researchers with regards to their work, and the implementation and accountability of journals, of funders, and of researchers in thinking about these issues. So there was 13 recommendations, but I'm just going to share with you today a few of the recommendations. The first recommendation that I want to highlight was the first recommendation. Researchers should not use race as a proxy for human genetic variation. This was one of the fundamental findings of this consensus study that clearly, as we think about genetic variation, that race is not a proxy or a surrogate for genetic variation, and that researchers should not use race as a proxy. The second recommendation was when grouping people in studies of human genetic variation, researchers should avoid typological thinking. This issue of how we use and communicate difference is extremely important, and typological thinking is an important component of that. Third recommendation I want to highlight is recommendation 6. Researchers should tailor their use of population descriptors to the type and purpose of the study and explain how and why they use the descriptor. This is fundamental with regards to research design, is the ability to communicate, to be transparent with regards to what you're doing in your study so that others can replicate it, as well as to understand how you're using descriptors within your work. So researchers, especially those who collect new data or propose new courses of study for data, should work in ongoing partnerships and study participants and community experts to integrate perspectives of the relevant communities and to inform the selection and use of population descriptors. One of the really interesting and important things that the study committee did was to articulate the fundamental importance of working with the communities that you're conducting research and to engage them in helping to think about how their populations are described, how terms are used, to get their input and to engage them in the process. And this too will help to create the nuance of understanding the complexity of difference in populations. A second area is with regards to communication to the media, a recognition that the scientist has an extremely important role of communicating their work and their findings with the media and the media with the public in a way to help to make sure that they are not reifying race as a biological construct or inappropriately having interpretation of their findings. And so the work of working with the media around dissemination is extremely important. So in summary, the areas of importance from the National Academies Working Group that was on populations, descriptors in genetics and genomics research is to avoid typological thinking, to measure environmental factors, that recognizing that not on all genetics is involved in looking at difference and health disparities and different outcomes, and engage communities and participants within the work. I want to highlight here, and I provide the QR code, that the National Academies has really a great set of resources that can be used to help individuals understand how genetic similarity, genetic variation plays its important role. I think these are great educational tools for the faculty members that are on this call today that can be used within your curriculum to help to explore and challenge students in thinking about these issues. But they also provide a way to provide some information in brief statements and brief fact sheets that can be helpful for individuals who are interested in this area. So what is NIH doing? Well, I just want to say that NIH has accepted the report and is considering the report with regards to policy. I just highlight here a blog that my colleague, Dr. Sherry Shuley, at All of Us and I published within Dr. Michael Lauer's blog about rethinking population descriptors in genetics and genomics research to make the extramural research community aware of our work. Our institute, the National Human Genome Research Institute, excuse me, developed a descriptor, an explainer to help individuals learn more about population descriptors. And I would encourage you to go to this website to learn more about that. So I want to shift, still working with the National Academies and the work that's been done at the National Academies. But I want to talk about a report that actually just came out less than 2 weeks ago. On October 30th, the National Academies published a new report, excuse me, Rethinking Race and Ethnicity in Biomedical Research. This is just a newly published report. An original report, the work that was sponsored by NIH, that was focused on scientific study design in genetics and genomics. And this new report that was funded by the Doris Duke Foundation is focused more broadly on biomedical research and clinical care. And you can see the QR code here that will take you to the report where you can download the report. But I thought it was important for me to talk about both reports now, that the original report on genomics that came out in 2023 and this new report that just came out in 2024, and how they really are consistent with regards to the guidance that is being provided to researchers. So the first recommendation in this new report is this: at every stage throughout the biomedical research process, researchers should scrutinize, evaluate, and decide whether the use of race and ethnicity is appropriate or inappropriate. And in publications, researchers should articulate their decisions and whether and how to use race and ethnicity in their research studies and reflect on the outcomes. This was a core recommendation that came out of this report that I think is fundamental and is consistent with the findings in the 2023 Population Descriptors for Genomics report. Second recommendation, researchers should operate with transparency at every stage in the development and application and evaluation of biomedical technology that may influence health and the use of race. This really gets to the issues of clinical algorithms and AI and medical devices, and how race has been used and how it's appropriate to use, and really helping researchers think about the potential biases and what are the issues that need to be taken into consideration with regards to use of race in these technologies. And the third area is that researchers should identify and partner with specific communities relevant to the research context. Again, this message of working with communities is an important part of the work. So this report identifies key considerations, and they use this really nice graphic to kind of think through the whole biomedical research process and the issues that you need to think about. And so the first area was about the use of race and ethnicity, and they articulate the need to disaggregate race and ethnicity, to define concepts, disclose the limitations in the approach of study design. The second area with regards to forming community partnerships and community engagement. They identify the importance of building trust, sustaining relationships, ensuring transparency, forming interdisciplinary teams, and understanding and respecting of study participants and study participants' engagement within genetics and genomics research. And then the next area with regards to inclusion and equity, they highlight that it's important to account for adequate time with regards to working with community, ensure equitable benefits to research benefits, and incorporate multiracial, multi-ethnic individuals throughout the study process. So to think about these issues. So there are multiple issues here that kind of at this intersect of thinking about the bioethical questions, the equity questions, and the study design questions, where I think this report can really help to lead and advance work with regards to population descriptors. And then the final area is to evaluate data sets and study methodology that's important to collect granular data, to go beyond our traditional categories of race and ethnicity, use appropriate categories in measurement, and disclose your limitations. Again, disclose your limitations of what you're doing. We recognize that all study designs have limitations and the importance of articulating those limitations. I want to make just one comment. You could say, well, I have an NIH study and I'm required to complete the inclusion enrollment report. And within the enrollment report, I'm restricted to certain categories. That is true, but there's also ability to do more. And so I just want you to know that the OMB categories are social political constructs. They are minimum set of categories, and they don't limit your scientific analysis. And I think that's the key issue I want to highlight here, that those categories don't limit in any kind of way the type of analysis. And so it's important to think about that when you're thinking about how they're used within your work. So I want to start to come to a closing with regards to my talk over the next few minutes. And I want to put this in context of where we are as a field and thinking about the history of race and genetics. Um, and when you look back over the field of genetics and you look back at how race has been used in a discriminatory and a racist, scientific racist way, it's been used with eugenics as a way of causing harm, that we must recognize today in 2024 the limitations and challenges that we must grapple with regards to a field. Excuse me. This slide from Alice Popejoy, again, one of my colleagues, has a number of images from over the last few years where in the public conversation and debate that people are thinking about this issue of use of race. And this issue with regards to how we interpret and talk about difference, how information from direct to consumer genetic testing about ancestry conflates with race. How do we think about the importance of race and genetics together? And that we're really at a point now where I think with these new reports, the two National Academies reports, where we are as we learn more about genetic similarity and genetic variation and the complexity of difference, that we're really at a point where we can take this information in a way to make a difference in how we present this information to the public and how do we design our studies and designing our studies to be much more thoughtful, careful, and transparent for the public and for our colleagues. So I would challenge all of us as we think about the field of genetics and genomics and its intersection with bioethics, that we conduct research over the next decade that really is pushing to explore these issues of identity, population descriptors, and genetic and genomics research, and how we move across this work in a way to actually improve health and improve our understanding of genetic similarity and genetic variation. One of my messages that I clearly want to say is that genetic variation exists and we recognize and it's important for health, but also how we communicate that is extremely important. So again, back to our strategic vision that we published in 2020, we included in that strategic vision 10 bold predictions. And these 10 bold predictions were predictions about what potentially could happen by 2030. And so these were areas that were both to be bold and to challenge the community, but also potentially could happen. And one of the bold predictions that we highlighted was this, that research in human genomics will have moved beyond population descriptors based on the historic and social constructs such as race. And I think now, as we come to the end of 2024 and looking back at the work that has been done by the National Academies and by scholars across the country and across the world, we really are moving and challenging everyone to think about this issue of how we see difference and how we move away from social identities and social constructs in race when we're talking about difference within the field of genetics and genomics. So with that, I want to come to my last message, and this is my take-home message. So when you go back to your office or to your class and you say, well, I listened to this talk, what was the message? The primary message is transparency. When you're conducting biomedical research, when you're conducting genetics and genomics research, being transparent in how you use descriptors in your studies, both for yourself and for your colleagues, but for the public. So with that, I want to say thank you. I want to highlight and encourage you to go to genome.gov to learn more about the work that we're doing at the National Human Genome Research Institute to study genetic similarity and genetic variation and the ethical, legal, and social issues that are important related to genetic variation and race, and to learn more about how your work may be incorporated within to this important area. So with that, thank you, and I look forward to our conversation.
Thank you, Mr. Bonham, so much for that talk. It's certainly given me a lot to think about, and I hope it's given a lot. In fact, I'm sure it's given our attendees a lot to think about as well. While people formulate their questions and submit them either to the chat or to the question and answer function, I thought I would take the privilege that's been given to me to be able to ask you the first question, if that's all right.
Okay.
So I wanted to ask about this concept of moving beyond that you just referenced, and how exactly is the best way to do that, right? So, um, is it the case that we need to, as it were, start over with our genomics research and genetics research, especially as it is applied to clinical practice? Or are there ways of interpreting or translating research that's previously been done with these less than ideal proxies in such a way that that research doesn't can still inform us even while not being the not adhering to these sorts of recommendations?
No, I think that's a great question. So first, this is an important topic that's ongoing of how do we um, use legacy data that we have that's extremely important, that may have descriptors that clearly we all think are inappropriate
Mhm.
and now in 2024. And our institute, in collaboration with several other institutes, held a conference in May of this year studying this question about legacy data. And so you can go to genome.gov and you actually can see the report from that conference that highlights some of the important research areas and some of the strategies. We see this as an area of research that's needed on the best practices for use of legacy data and a recognition that this is one of the challenges that we have. But there's no expectation that you're not going to use legacy data.
Mm.
We need to continue. We've developed resources, we've built resources, we've had collaborations with communities that go back years and years, and we have their data, and that it's important that that data is part of research going forward to improve health.
Mm.
But we do need to understand and be able to make modifications in how we talk about and describe populations. So there's a lot of work to be done, both from a research perspective on this, but there's also been a lot of thought. And so I would encourage people to go to that report to learn a little bit about that set of scientists that came together to help us think about these issues. But we see this as a high area of importance from future research that's needed.
Thanks. That's very helpful. I'm very glad to hear that this is already being thought about, and that in fact there's a report I can go read and have some of my questions answered. Let me turn to some of the questions that have been posted in the chat. So first, from Jahan Benamu, thank you so much for this important presentation. When studying complex traits, do you think that previously identified SNPs or variants may have been overestimated, since these studies don't control for key social determinants of health? If so, how do we take this into consideration when thinking about using genetics in clinical practice? Thank you.
That's a great question. I think that the potential of over uh, basing the differences that we see based on genetics is real, and that we need to be very careful and thoughtful. And as a clinician, it is very important that you recognize that the variations and the differences that we see and risk differences that we see are not necessarily just genetics. And that's why it's really important in new studies that you're including environmental measures and social measures within the analysis, that you're conducting both genetic and genomic research with social context and environmental context work to better understand the causes of differences that we see with regards to risk. And so, as an individual clinician, I think it's really important for you to always think about that individual patient in front of you and that one individual, and trying to understand what may be important with regards to their risk and disease. And clearly, understanding their genetic background is an important part of it, but it's not the whole component. You must be thinking about those clinical, social, and environmental issues that are important in individuals' health.
Great. This question comes from my co-fellow, Dr. Wong. She writes, this question is about the importance of engaging communities. How can we engage communities or individuals who resist the claim that race is a social construct and therefore should not be used as a population descriptor? What are the potential costs, in particular to genetics and genomics research, of failing to engage such communities or individuals?
Well, I guess my perspective here is that we might need to make sure that we engage all communities and seek to bring them into helping us think about study design and how we describe our population. So if I understand your question correctly, you're saying, there are communities that say, well, race is important, and I want my population, or I want to be identified based on a racial identity in this genetic study. I don't care what you say or what that report from the National Academy says. I think it's important to understand what the community wants. And I think it's important for you also to be clear and provide nuance in your reporting. So that ability to kind of both hear the community, work with the community, but also provide complexity to how you describe the populations, I think is part of your role as a scientist. And so I think that ability to continue to work in that kind of way, building those community partnerships over time where you're creating a stronger relationship, where clearly a community is then listening to you and you're listening to the community, I think is really important part of the work.
Dr. Finder from Cedars asks, can you offer examples when using race and or ethnicity as part of genetic studies is appropriate?
Yeah, I think if you're conducting a study that is looking at differences based on a social context. So let's say you're conducting a study that has both a genetic component as well as understanding how maybe geographic location or some social context of an individual's life, where race is really the important context, that you use those terms also. So that would be an example when you think about health disparities research. racial and ethnic health disparities research, that they may be important because of the type of work you're doing, that you both talk about genetic ancestry or genetic similarity, but you also talk about individuals' self-identified race or ethnicity, because it's important in understanding the research question that you're looking at. I think the issue is if the only question you're asking is a genetic question, then the use of self-identified race and ethnicity may not be the most appropriate. But if you're looking at genetic questions or environmental questions and social questions together, which I argue is an important thing to do, then the ability to use both descriptors can be extremely important.
Great. Dr. Hines asks, the concepts of race and ethnicity are embedded in the infrastructure of research, as we sort of previously discussed. Can we move beyond these categories on a voluntary basis, or is there need for governmental oversight and enforcement? What would that look like? So essentially, how can these recommendations be implemented in research?
Yeah, I would love I would love the question, what their opinion is on what the best way to do this. I think as someone who is really involved in helping to think about, okay, what do we do? How do we engage and work with researchers and scientists to help to really challenge these issues? A number of professional societies have gotten engaged in this conversation and are participating and thinking about these issues. I did not share a slide today that I shared yesterday from at Cedars-Sinai, where there has been a group of journal editors from very high impact journals that came together and came up with 10 principles that they think are important for the use of race and ethnicity in their journals. And this included Nature, New England Journal of Medicine, and JAMA, and it really articulated what they thought was important areas. So I think there's a complexity of issues here of how we can move forward. Should there be some government mandate? Um, um, my own view of this is just my view, is that I think that as a scientific community that we really want to develop norms and customs that are based on the best science, and that the peer review process of reviewing both before journal articles and grant reviews is an important component of that process.
Excellent. Dr. Rejo asks, can you say a bit more about the harms to individuals or groups if we fail to address diversity in genomics databases? What kind of risks are there to individuals or groups in terms of clinical impact? And just to add a little bit, I'm wondering if you can speak to that from your own research with sickle cell, for instance, if that's one of the areas in which that
Yeah,
crops up.
yeah, no, I think that this is an extremely important area, and it comes up in multiple areas, and individuals who are on this webinar can give me examples. But I'll talk about the sickle cell. But before I get to the sickle cell, this recognition when you get information back, now that genomic data is part of clinical care, and you sequence an individual's genome, you do an exome or you do a whole genome sequencing of an individual patient, and you get these variants back and you don't know what they mean. So those variants of unknown significance, and you're trying to make a decision about someone's risk for cancer or risk for heart disease or risk for something else, and you just don't know because we don't have enough information about individuals of that background. We have seen a number of examples where we might identify someone and a variant being pathogenic, but it's because we didn't have enough information to find that that's a very common variant in that ancestral population with no risk. And so that is a major fundamental issue. It comes up in a variety of different areas where we just don't know. We have some piece of information and you want to share with your patient how that information is important to their health, and you don't have the ability to do that. With regards to my work with sickle cell disease and some of the things that are going on there, the exciting work that's going on with the new gene therapies and the curative therapies that work with many patients, but not with everyone well. And some people are at higher risk for potential cancer. Well, why is that? Is there some genomic component of that we just don't know yet? That we're still learning with these new gene therapies about what populations within the sickle cell disease population may be at higher risk and those that are at lower risk where these treatments are most appropriate. So we're just learning a lot. We're still at this early stage, and without additional information, without additional people participating in research, without diversity of ancestral backgrounds, we just don't have the complexity of information to be able to make good interpretations about risk with regards to these new therapies.
Sort of following up on that point, an anonymous attendee says, "Thank you so much for your excellent talk. Your work has been so interesting. As a researcher in sickle cell genetic research, something we have been struggling with is enrolling patients from marginalized communities onto genetic research protocols, with medical mistrust playing a role in hesitancy to participate. Do you feel that involving community partners are the strongest way to increase recruitment rates, or are there other avenues that you think could increase willingness to participate and promote diversity and equity in genetic research?"
Yeah, no, I think working with community organizations is essential, and building that kind of trusting relationship, and the ability for groups to make their communities aware of opportunities so they can make that decision whether they want to participate or not. In my own work, we've collaborated with communities, sickle cell disease communities across the country around what are the issues of importance to them. And so I think that's fundamental. But I also think how you build your relationships with your individual patients, and that trusting relationship that you build with your patient as a provider and then also as a clinical researcher is extremely important. So I think it takes all those things, clearly working with advocacy organizations, but also being recognized as someone who is truly interested in that patient population, is engaged in providing care or supporting them in whatever the most appropriate way for your work is extremely important.
Good. I'll just note that since we're under 5 minutes, if anyone has additional questions that they want to add into the Q&A, now is the time to do so. We do have one from Marwa who asks, "How is ethnicity determined prior to studies? What are the options for participants who do not know their ethnicity or who do not have a clear image of their genetic history and primarily identify themselves on the basis of race?"
Yeah, no, that's a fascinating question. So, one of the things that I've explored over the years is how health professionals understand race and ethnicity. And we did a survey of general internists across the country, and we asked them the question, what does race mean to you, and what does ethnicity mean to you? Two separate questions. And we found that these health professionals, and these were general internists, that over 80% of them identified race as a biological or genetic group. And when we asked the question of what ethnicity means to them, over 60% identified ethnicity as a cultural group. And the reason I give you that example is that these terms are not set in stone in how people understand and interpret them and what they mean. And so when you ask the question about how people determine their ethnicity, I think it's really all the context of what does it mean to you? Is it religion? Is it geographic area? Is it cultural background? Is it genetics? How do we understand what that means? And that's why I think it's so important when you're conducting research, and let's say you're doing a study and the issue of ethnicity is an important component of it, is to explain what that is for your study and how it's being used in that study versus another study, so that there's a clarity of understanding that. But these terms are not, single views and opinions about what they mean. And we must recognize that. And that's why it's so important that we're transparent in what we are doing in our work, that we explain, well, when we say race, this is what it means, or when we say ethnicity, this is what it means, and how we are applying it to the study that we're conducting.
I think,
So, Vince, we are actually more or less at the end of our time. On behalf of the Ethics Centre and the health system, I really want to thank you for an extremely thorough and very thought-provoking presentation, and also for your willingness to engage with questions so adeptly and also in such depth. So again, on behalf also of Tom and Leslie, the sponsors of the event, myself and my colleagues at the Ethics Centre, and everyone. We have a much larger turnout than we anticipated, thanks in part due to both your own prestige and also the wonderful advertising skills of our admin, Hamist. So hopefully at some point we can have you here in person and we can engage with groups of researchers and clinicians in person. But until that time, again, thank you most sincerely for your presentation today.
Great. Well, and thank you again for the invitation.
Okay, great.